experience. It's like a doctor will diagnose a patient, and they will give a formula, the herbal decoctions to the patients based on their situations. From the herbal formula, they always to come from the whole plant, fruit, seeds, and flower. This is this herbal decoction is a very beautiful and ministry of the TCM are synergistic and multifunction. However, we can have meet a challenge in the TCM development is, for example, the multifunction, synergy, multi-pathway, multi-target, and the compound constituents. Now, in our study, we will give a example is the downgrade Shanghai DBD. Why we use these formulas? First, the formulation pharmacy, Li Dongyuan, they create a formula is use the astrogalic radius AR one C with the agenesis synthesis radius ASR downgrade with pipe form 5 to 1 ratio and bone boiling 2 hours to form a hot water consumer decoctions. And so in the literature is claimed that the woman's disease such as the cures such as the menopausal disorder and other diseases such as the cardiovascular functions. In the lectures, the DBT can emphasize DBT can treat menopausal disorders by lowering the qi and enrich the blood. In our laboratory, we have do a lot of study about the biological function in DBT. For example, anthropoetics, estrogenesis, osteogenesis, cardiovascular, and anti-oxidative functions. So we will, in order to modernization the DBTs, we do a lot of study to use this very scientific approach to understanding the literatures. For example, we will do a lot of research for the why the AR and ASR is 5 to 1 ratios, and why the ASR is rostered with the envelopes and the extraction method. It is the reason is in order to optimize the active ingredient as well as increase the biological functions. However, there have some of the black box we cannot understand. For example, DBT can claim that can enrich the qi. And what is the qi? It's easy to understanding. It's not easy to understanding. So nowadays we use the synthetic biologists to solve this a lot of the complicated questions. They have a 3 elements: biology, computational, and technologies. Give the new insight, new hypotheses, and new biological questions to different very complex situations. For example, the COVID-19, the developmental biologies, neuron disease, as well as the environmental pollutions. Nowadays, for the systematic biologists, we always to use the multi-omics technologies. What is the omics? Is the collect the whole set of the uh, biological molecules. For example, in our study, we use the mass spectrometry to detect lots of the lipids, metabolites, and the proteins. Here, in our study, we will try to use the omics technologies to solve the questions and or the black box of the DBT. The first question is, DBT can reach the qi. So what is the qi? We use the method first to resolve these questions. And the second ones, they have a lot of active ingredients. What is the roles of ingredient and the measurement mechanisms? And the third ones, in the DBT, they have the proportion of the AR extract is very high proportions, and AR is the very attribute on hot stimulations. What is the role and action mechanisms in the AR? In our study, we would like to solve it. So the part one, the effective of the DBT to mitochondrial bioenergenesis. So we will use the we will extract the ASR, AR, and DBTs water extract for 2 hours, and the extract will go to the fish dryer, and will be suspended with some solvent to put in the HPLC for the QC and QA analysis. So, and we use the dye-away detector as well as the evaporative light scattering detectors to detect the active ingredients. Glycosin from melting astrogalsite 4 is the major chemical in the AR. The fluoric acid and the isophone lingostildide is for ASR extract. And then we would like to examine the anti-oxidative effect of the DBTs. So we will use the total phenolic analysis and the vertical scattering assays to understand the antioxidative functions. And also, we will use the cell line models. For example, we'll use the TPHP. TPHP is the can provide the cell have a oxidative stress, and if treat different kind of stress, they can prevent the oxidative stress. And also, and now we will use the very new techniques is the seahorse metabolic fluxomics to detect the concentration or change of hydrogen ion or oxygen ion. In these situations, we will use the mitostress assay to add the 3 kinds of inhibitor to to get the 6 parameter: basal respirations, ATP productions, protein leak, and maximum respirations, spare capacity, as well as the non-mitochondrial respiration to understand the mitochondrial bioenergenesis. First, we will the cell line use the H9C2 cell lines. First, before the treat the drug, we would like to optimize the concentration of the FCCP as well as the cell density. In our experiment, we'll optimize the cell density is 5,000 cells per well, as well as FCCP is 3 micromole. So, sorry. Okay, so we will the H9C2 cell line treat the different concentration of the extracts. All extracts show the metabolic function is dose-dependent manner, and the DBT process the strong metabolic effects to basal respirations, protein leak, ATP production, as well as the non-mitochondrial respirations. So we would like to more a situation is if some cell is in a sick or disease stage, what will happen? So we'll also use the TVHP as a oxidative stress. And first, we will be treated with the herbal extract for 24 hours and exposure to TPHP for 3 hours. And also, our result can show that DBT could improve the metabolic ability in stressed cell. For example, the basal respirations, protein leak, ATP production, as well as non-mitochondrial respirations. So in order to understand the role of active compound in DBT for the mitochondrial biogenesis, so we study these five compounds. The result can show that the astrogalside 4 process the strong metabolic effects to basal respirations, ATP production, as well as the maximum respirations. And the linguostilda is contribute the strong metabolic effect to the protein link. In order to further to investigate the more upstream level of the molecular functions, so we employ the mitochondrial membrane potentials to to investigate the the ability of the ATP production. So the result can show that the DBT is treated after treated with DBTs, the mitochondrial function is maintaining a stable state even in the health cell model and weak respiration cell models. Also, we'd like to investigate the mitochondrial biogenesis and mitochondrial DNA replication related genes. And the result can show that most of the after DBT treatments, most of the mitochondrial biogenesis related genes is upregulated more than other treatment group. In order to further confirm the mitochondrial morphology improvement, we will use the mitochondria to sting the mitochondrial network. We can show that the mitochondrial intensities, the average parameters, for example, the circularities is down is decreased, and also the the longage is increased, so they can improve but the morphology of the mitochondria, as well as we will use the electron microscope to confirm the result of the confocal microscope. This is consistent with our results. So for the energy metabolism part sub-conclusions, we conduct a comprehensive analysis of the effect of DBT on mitochondria, including enhanced antioxidative effects, moderate the mitochondrial bioenergesis, stabilize the mitochondrial membrane potential, upregulate the mitochondrial biogenesis genes, as well as improve the mitochondrial morphologies. Second one, we'd like to use the integrated omics to dissect the synergistic effect in DBT on cultured osteoblasts. The story is that the gargosine is a major and very high abundant compound in the DBT. So we first in separate by the HPL, the semi preparation column to separate the gargosine fraction, as well as so the DBT lockout with gargosine and the gargosine fraction is formed, and the result can show that the gargosine is lockout, DBT lockout is successfully to removed the most of the gargosine. So we use first, then we use the gargosine lockout to screen different biological function. We found that an interesting thing is the osteogenesis. DBT, the compared to DBT lockout gargosine, is the DBT lockout gargosine reduced is most reduced compared to the DBT. However, the gargosine is not less or no any effect. It's very tricky bonds. So we found that there have a phenomenon that the gargosine in the osteogenesis is not not an active compound, but you can define a acoustic compounds. It's like a orchestra. If you without the conductor, you cannot play any music. For the deep into detail to see these functions, so we will first. I would like to introduce the bone remodeling process. First, we the formation is based on the hemotopic stem cell to form the osteoclast, as well as mesenchymal stem cell to form the osteoblast precursor cell to form the osteoblast. After the equilibrium, they will. This is the bone formations. In the bone formations, they have a three-stage: proliferation, differentiation, and mineralizations. And if try to use glycosin, the glycosin, the ammonia density about the 1619 nanogram, and so is these concentrations, you can see they have not any result and not any effects about the bone proliferations and ALP activity. It means that the differentiations as well as the run X luciferase assays. And the second data, we can show that the glycosid knockout was shown to reduce the osteoporotic effects compared to DBT is reduced. Uh, so we need to do the cohesion and the adhesion ability. The glycosid knockout is much more decreased compared to the DBT. So, and to further use the ex vivo model to confirm our hypothesis, the we will use the micromass cultures to stimulate to test the osteogenesis. We can find that if this is the delta DBT logouts, we can show that the DBT is much more density cellulose. It means that they have a strong osteogenesis compared to DBT logouts. This shows that the DBT glycosid logout is very effective on the on the osteoporotic functions. So we can imagine a diagrams if this is the DBT systems and glycosin is control all the compounds in the DBT system. If without the glycosin, they this is very the DBT system is mixed up and per, is mixed up and cannot do and destroy any functions. This is butterfly effect. So how to solve these very messy situations? We've used the omics, multi-omics. If single omics, just some bright people to touch touch the elephants. Or this is friend, this is a trees. But if use the multi-omics, we can see the whole mixture of the DBT. So first of all, you use the transcriptomic analysis. We can to compare to DBT and DBT logout. We to detect a lot of set of mRNA and non-coding RNA. First, we found that the mRNA, the very significant changes is 337 3P and let 3P 5Ps, which is the very important for the osteoporotic functions. And also, we can compare with the DBT and DBT logouts. We can find there has five different major pathway is affected with compared to the logout DBT. For example, we will the DBT is more mRNA metabolic process, and the mRNA splitting. And the second one is the amino acid metabolisms. And the third one is the lipid metabolism. Third one is energy metabolism, as well as some function related to bone formation. For example, the magnesium ion bindings, as well as the estrogen dependent gene expressions. It's very critical in the bone formations. And interestingly, we can find that there have some changes about the immune response. It's very interesting. However, we can find a one thing is not a lot of pathway is not related to the bone formation or bone differentiation directly. So we employ the proteomics approach with the cell, the osteoblasts treated with different herbal extracts. The protein was extracted, and after the tissue digestion, we become to a peptide. The peptide will introduce to the nano LC orbital tracks for analysis. They will detect the peptide, the whole chain of peptide. After the collision, they can know that the amino acid sequence and use the computational approach and the peptide spectrum matching can predict which peptides and predict which protein we detect. And our quantification method is the label free quantification to calculation the p area of the precursor ions. And the statistic you use the p-value is smaller than 0.01, and the floating is lower and higher than 1.5. So this is the very general principles. When see a peptide, and the peptide will collision and give the amino acid sequence, so we can detect which peptides. So this is the result, and we can find the different peptide. The peptide is past the 1% false discovery rate, and we can find about the 2,000 proteins, and the significant the defensive expression proteins is about 200. Okay, and so in this proteomics level, we can find that a lot of pathways, for example, the wing signaling and collagen biosynthesis, as well as the cell junction cytoskeletons, is very directly related to the bone development is appeared. And also some pathway or mechanism, for example, the RNA metabolisms, energy metabolism, as well as lipid metabolism and glucose metabolism appeared in in protein level as well as in the RNA levels. And also we can find some novel approach. For example, the novel pathway. For example, the zinc ion transmembrane and the launch receptor process. And they can give a lot of information about how to deduce the pathway. So we will use the level analysis to we can result and get the forecasting. And this forecasting can more to know the what happened will not out the glycosins and compared to the DBT compared to glycosin, the DBT lockout glycosin. We can find that the DBT can increase the launch pathways and AKT pathways, and also the heat shock proteins. This is a more low custody we load and we dissolved. And in next steps, we'd like to find, do you have any the post-transcriptional modification, translational modification effects about in the proteomics analysis? we can find that the most a lot of most changes is the acylated proteins. And after the pathway analysis, we can find that very important key changes. For example, the actin cytoskeleton, osteoblastic differentiation, as well as collagens. So so when the DBT without the cargosine, they have a very important changes in osteoblastic functions. Now, with because in the RNA and protein level is find a lot of mechanism or pathway about the lipid in order to further study which lipid is changes and why it changes. So we'll do the lipidomics. It's very simple. It's just the use the cell is treated with the extracts and extract our metabolites or lipids and go to the inject to the LC and MS. So after they can have a MS and after correction have a MS specials. After alignment, we will do the multivariate analysis to find the significant change of the biomarker. After the compound identification can guess which pathway is triggered. So we first will find this in the PCA analysis. This is a QC samples, and this QC sample uh, is more important for the metabolomic analysis. And in the first in finally we will use the OPLDA pathway to find the difference and use the variable in projection uh, the features to find why these two groups are separate together is based on these features. So we'll use different broker database. We can find that very easily to find that they have a two group are separate. You this the red one is the DBT, the purple is DBT logout, and the compared to DBT compared to DBT logouts, we can find the group of the, for example, the fatty acid, fatty amino acids is a decrease. However, the eicosaccharides and sphingolipids increase the expression compared to the DBT logout. So finally, I will use the integrated omics analysis to compare our results. Is our relevance. You use the RNA, protein, and lipid data to compare to each others to verify our omics results. We can find that this is very similar to our RNA protein level. For example, this is related to RNA metabolism. This is related to the osteoblasts and enzyme metabolisms. And in order to find the more local information, local pathways, we will again put the RNA, protein, and metabolite data to the level analysis. We can find that some local protein, local pathway are triggered. For example, the hypoxia-induced factors and the VEGF and mitochondria complex 1. The signal is upregulated DBT compared to DBT logout. So they have a lot of pathways to determine. So we would like to do some biological assay. For example, we use the first we'll deduct the ROS signal. So we after treat with different herbal medicines, we can find that for the ROS assays, it's very important to the bone formation, as well as ROS signal can induce the AMPK, which is related to the biogenesis. And also, the DBT glycosid is not out, is, is more decreased compared to the DBT. And also, we can, again, we'll find you examine the mitochondrial mapping potentials. So the mitochondrial potential is, interestingly, is low any statistical significance between the DBT and the DBT glycosin log. So again, because the protein level at the lipid level is changed by the is changed is changed by the DBT compared to DBT logs. So we use the also to perform the seahorse analysis. In this time, we investigate the oxidative phosphorylation as well as the glycolysis. We can find that the major difference is in the between the DBT and DBT logout. The degrees of DBT logout is statistical significant is in the maximum respiration spare capacity, glycolysis capacity, as well as glycolysis reserve. So we will use the electron microscope to confirm the result of the mitochondria biogenesis. So we'll after the EM quantifications, we will find that the mitochondrial air per cell area, the mitochondrial area, as well as the length, is much more statistically significant decrease compared to DBT. And also, finally, we will use the uh, mitochondrial rat to detect the mitochondrial dynamics. It means that to is to more investigate the fusion and fission, and so that the even after the judgments, the DBT log compared to the DBT is much more reduced in the biological branch, level branch length and footprint. So, with summary, we can propose the action mechanism will mainly focus involved in the glycolysis metabolism, lipid metabolism, brain pathway, PCG1 alpha pathway, as well as the hypoxial response element pathways. This is a major pathway to is regulated by the glycosin. In the sub-conclusions, which is demonstration the in-depth and integrated peptides to preserve the active glycosin of the DBT in osteoblasts. The second one is reduce the osteoblastic functions of the DBT log of in cultured osteoblasts compared to DBT. The third one is osteoblastic function of DBT is not related to glycolysis, energy metabolism, AMPK pathway, lipid metabolism, as well as immune response through the integrated omics approach. The results suggest that the glycosin can engineer control the osteoblastic function and the pathways in DBT naturally and spontaneously. The third part is the serum metabolomics and lipidomics to verify the thermogenesis of the astroglycopractice on this induced animal model. So first, we'll use the mice. They will treat it with this model for information, and after this, we will the the mice model will administer different drug extracts. This is the hot stimulated drug extracts. This is cool stimulating drug extracts, and the serum you extracted and extracted some metabolites and lipid classes to introduce to the triple time of flight mass spectrometry for metabolomics and lipidomic analysis. For the free and non-free fatty acid, we'll introduce to the GCMS. So for the temperature change of this bottle, we can show that the astroglycated and collagenous serum will show the opposite effect to the body temperatures. And we can find that a interesting things at in the after the 8 hours is the critical reference point for the temperature changes. The second one, we will use the fatty acid to this is the fatty acid was extracted and will identify by the GCMS. And the free fatty acid, the acidified fatty acid, will put it into different model, PCA, PLSDA, and OPLSDA model for the classification. We can find that the 8 hours can separate the hot stimulated herbs and the cool stimulated herbs. So in order to know more about the detail about the 8 hours, we can use the heat map as so different content, the difference amount and the flow change compared with the hot stimuli and cold stimuli hubs. We can find that generally the hot stimulating group compared to cold stimulating group is the fatty acid is uh is less is much more decreased. In order to investigate the more about the lipid changes, so we appoint the lipidomics, and the result is so that they use the heat map for the presentations, and the major change about the difference about the hot stimuli and cold stimuli has is the triacylglycerol as well as the sprinklemycin is much more decreased in the hot stimuli group. So we would like to more about the to investigate the dimension of the metabolomics, serum metabolomics. So we'll use to extract the metabolites and put it in the time of fine mass spectrometries. And this metabolite was identified. And also, we will put it in different statistic models. We can find that the 8 hours is also a good separation with the hot stimulating herbs group and with the cold stimulating herbs group. So the features was identified by the OPLSDA and the scoreboard, and we can find the like about near to the 100 the different metabolites. We can find a different group amino acid, the boric acids, carotenes, PEPIPS, lyso lyso phosphatidylcholine and phosphatidylphenolic amine and the phosphocholine. We can find a tricky and very tricky tragedies and very patterns for after treated with the hot stimuli helps the amino acid, and this kind of amino acid group will decrease the concentration compared to the cold. And this is related to the immune response and thrombogenesis metabolites. In the this is the lipid group, and also is always to increase. And this lipid group is a, as a messenger for immune response. So finally, we'll involve you use the metabolite level analysis to give some more information about the upstream levels. We can find that in the hot stimulating groups, you can increase the expression of the P58 MAPK pathways is related to the informations. And luckily, the co-stimulation group is decrease the N-alpha kappa B pathways. It's very interesting the observation. Is the hot and cold stimuli is most likely related to information. So in the sub-conclusions, the yeast induced information model after cold and hot herbal medicine were used as a model system. The multi-omics approach identified the customary of the cold and hot treatment, the red syrup, by using PLSD model, which is identified the 8-hour treatments was metabolite perturbations points of the herbal induced thermogenic regulations. The third point is the levels of the metabolites in the serums. For example, the lyso-PE, lyso-PC, and so the positive relationship with the regulation of the body temperature and the levels of amino acid fatty acid were more negative correlation with the temperature change at 8 hours. So in my overall findings, the low-flow, robust, and high-throughput integrated omics approach was performed. And the second one, the platform can contribute to our understanding of primary mechanism of DBT, such as energy mechanism and the thermogenesis. And the third one, our approach can serve the critical backdrop for the further study that characterization the impact of the broader array of the different factors of the herbal medicine. Finally, the investigation of the action mechanism in applying the multi-target drugs or combination therapies should be necessary for the modernization of the herbal medicines. Finally, we will thank my supervisor, Professor Carl James, and Professor Harry Lam, advising committee, Robert Chi, Warren Song, and defense examiner, Professor W.K. Wong, Professor Towen Bell, and Professor Dirk Ben, and special thanks to my colleague and the fans. And any questions are welcome.
Tasha, how many minutes?
43 minutes.
43 minutes? Tasha? Wait, Tasha, over.
It's 43 minutes.
Okay. So let me ask somebody. So Aline, can you turn on your audio?
Yes.
So if Kenneth giving this seminar, do you what grade that you give it to him?
What grade? I don't know. I don't have enough knowledge to judge his
No,
PhD.
just as a student, what grade that you will give it to him?
Presentation points. I think I'd give him like
everything. So don't
everything.
waste your time. Just give a grade.
B plus.
Huh?
B plus.
B plus. Okay, very good. So you give a B plus to Kenneth. That means you understand totally what Kenneth is talking about. Can you tell me now?
No.
So you don't understand. You give a B plus. Right?
Yeah.
So do you know? Do you understand what he's talking about?
I think I understand like most of the parts, but I wouldn't be able to explain. Yeah. So
let me talk about the phenotypic. So what is the outcome from that particular study?
So from that particular study, he did find that it's the last part, the cold and the hot part, right? So
the lipidomics analysis.
Although the part before that. Yes, no. I know what he did, but I don't know.
So regarding the last part, the hot and cold. So what is the final outcome? What is the major input that you think coming out from that study?
I think the difference was he found that the difference between the hot and the cold Chinese medicines was that they regulated different, they upregulated and downregulated different lipids and inside the body.
Such as? So you give a B plus, So Ali, you are very generous. So let me ask Gary. So which grade that you keep?
Maybe B minus.
B minus. So that means that you understand Kenneth's work a little bit at least, I suppose.
Yes. Yes.
So what is DBT lockout?
DPD lockout is the lockout of a chemical color coding. Is it correct?
Yes, that one that you understand that. So what about the mitochondria with the DBT? What is the outcome that he thinks that you can get it from his study?
I can only remember the ROS is decreasing.
ROS is decreasing.
I can only think of the elongation of the mitochondria.
Increase or decrease?
Increase.
Okay, what else?
And then overall function of the mitochondria is better.
So it's not very specific. So sure it's better because it's the work that he is not able to get that thesis out. Okay, Queenie.
Yeah.
What is your grade?
What is my B range?
So B range. So I think you are very generous to Kenneth. If I were one of the students, I would give fail to Kenneth. All right. So Kenneth.
Yes.
Unfortunately, you are the one that are having the PhD exam for so many new students sitting in this room today. All right. So including we have Gaga and Chen. All right. So and also Nan and also Sa. So you are setting an example that will fail in the PhD exam. All right. So I just want to remind you that if everybody present like what Kenneth did, so most likely the examiner will give a hard time to the student and also maybe give a fail to the students. Kenneth.
Yes.
I have been writing the paper with you together, but still I do not catch your presentation. What are the focus? All right, so you have a different study and what are the focus? I kind of not able to get it out from your talk. So I do not understand your English is not, well, where you graduate, Kenneth?
Okay.
Where you graduate? What is your undergrad study?
4 years.
Where?
4 years ago.
Huh?
HKUSD.
So HKUSD students have a very poor English presentation. All right. So I just well, this campus is going down because of you, Kenneth. All right. So the only thing that you speak a lot is so. Each sentence you have so. All right. So I do not understand what you are presenting. So the point is, when you give a slide, you need to tell me the focus. What is you want me to get from your slide? And you are very lazy. I give you one example. Show me some of those analysis, any one of the slide. PCA and DLS, whatever A. So if the end of the presentation. Go further down. Okay. Go the other one. Yeah, go further down. Yeah. The PCA, you have done a lot of those PCA analysis. No, Kenneth.
Yes.
So can you give me the slide? So
PCA analysis.
okay. No, not this one. This one is better.
This one.
Okay, let's say a 50. So you are very lazy. You just copy the figure, the diagram from the paper, and then you just keep a talk. So what is A? Let me ask Gao Xiong, what is A? Do you know? Gao Xiong, where are you? Gao Xiong. So what is A? What is A?
It's the figure A shows the PCA analysis
B,
of B.
C, D.
No,
So Kenneth.
Hmm.
So I am the one writing the paper. Maybe I understand what is A, B, C, D, and D. So what is the meaning of A, B, C, D, and D in this figure?
A is the PCA, B is the PLSD,
Oh,
C is OPSD.
Oh, wow. Now you know. So do you have a figure legend for presentation?
Yes.
You have a figure legend for presentation?
No.
So what you will do?
So put the lamps in near the figure.
Okay, so this is also, let's look at the 51. 51.
51.
Okay. So this one, and again, so the NS, what is NS? Amy.
Hello.
So Amy, what is MS? Do you know in that diagram? What is this diagram representing for? Do you know?
Maybe I'm not very.
You don't know. Okay, that's good. All right, Queenie, do you know? What is the diagram corresponding to?
I think this is a heat map showing the changes of the fatty acid after using the GCMS to analyze the
Okay, so what is the NSCS?
MSCS. The abbreviation of those herbs.
Okay, so is there any abbreviation in here? Kenneth, do you again have a figure legend in here?
No figure.
So what you need to do?
The full lamp. Add the full lamp near to
So
human.
you have plenty of space in this figure, all right?
Yes.
So do not just, okay, give me another example. Can you go further down? 52, 53, for example. All right, same again. All right, so what is A, B, C, D, E? Nobody know. All right, so 52. Okay. What is A, B, C? You don't know, right? Kenneth.
I know.
So at least my student do not know. So
Okay.
I can ask any one of them in here. So Becky, do you know what is A, B, C, D?
I don't know, but I suppose they maybe the PC and then.
Wow, nobody know. All right, so Kenneth, don't be so lazy. So on the right hand side, that heat map. So I need to add another Evan.
Yes.
So can you tell me those heat map in here? Those are red and blue color. What is this corresponding to?
I'm not sure.
You're not sure. So let me ask Xiao Zha. You don't know,
Yes.
right?
I'm here. I think it's about the full change of this, the effect of her.
Okay, the change of all those, this is lipid, right? So which one is which? Which one is DBT?
I don't know.
No?
I think he should label the name for.
Kenneth, you have space. So I know that on the left-hand side, you have abbreviation for the color, right?
Yes.
But do you know that that color is the smaller size that you can never use?
Okay.
If I were you, I will put the name right on the top of the clustering analysis, right?
Okay.
Or make the color abbreviation much bigger.
Okay.
So besides, I have many complaints regarding your seminar. Number one, your slide is poorly done, like what I just mentioned. you do not make full use of the space. Let's look at the conclusion. Okay, 57. All right. So one thing is, Kenneth, if you want to highlight the wording in here, for example, integrate omics platform, is that what you want to highlight?
Yes.
So I will not use purple or red color on the black background. Why?
Because the contrast
It don't come out. If you want them to come out, which color that you use? Let's ask Kelvin. Kelvin, which color that you think that you can use?
Should be yellow or some very
green,
bright color.
yellow, right? So the other thing is you have 1, 2, 3, 4, right? So if I were you, Kenneth, do you know how to make good reference in the paper?
Good reference.
Right, reference. I have been working with you on the many times on making a paper reference, right?
Yes.
So how to make a good looking reference?
The style is consistency.
Look at this one. Can you look at them? Okay.
Make use of the space.
No, it's not the space. You have the name and then you have a tab. Do you know what is tab?
You mean the
Kevin, can you help my friend Kenneth?
Yes,
Kevin.
no problem. Yeah.
So number 1, you put down lawful, robust, and high throughput integrate omics platform, proteomic, metanomic, and metabolic flux. So and then the one that with the metanomic that work, you put a tab.
Okay.
So look at the reference, Kenneth.
Okay.
All right, my friend.
Okay.
Okay, so I think your English is very poor, Kenneth.
Okay.
It's not okay. You keep on saying okay, you're just asking me to leave, right? So your English, you do not practice. So for all this week, you have been very busy. I think that you have been working very hard with your slide and make a very good presentation. Apparently, it's not. So, Gary, so what else you think that Kenneth can improve in his English?
Maybe the grammar can be improved for some description wordings like decrease and increase. I think he can say more clearly.
So the English is not like when you come out a slide, you need to get a major impact from the slide for us. First of all, your presentation is not for Kenneth Guan. Why? Kenneth.
For the examiner.
For everybody, in particular the examiner. This is the most important. So if this is only for Kenneth Grant, we can all go home because you are talking to yourself.
Okay.
So when you have a slide, you need to tell me what is the focus of that slide that you want me to look at.
Okay.
So don't come out okay very fast. So if this is a come out okay very fast, it looks like a reflex. So do you know what is a reflex?
Reflex.
Reflex. Muscle reflex.
I know.
You studied in neurobiology?
Yes, I know. Spontaneous action.
Spontaneous action. That means everything Carl Jim says is yes, yes, yes. And never the signal go to the central nervous system. Never go to think.
I got it. Yeah.
So you don't got it. All right. So you still respond very fast. It looks like a reflex. All right. Okay. When you speak English, can you speak it with a tone? All right. So you look like if I have to say that in Cantonese, 好似南摩咁嘅嘰哩咕嚕嘰哩咕嚕. So I saw that everybody falling asleep. All right. So Alim fall asleep, and Anan falling asleep, and my friend Maggie is falling asleep also. All right. So be So who did the work, Kenneth? Kenneth, who did the work? Kenneth Grant, right?
I did the yes.
Okay, good. So who should be more excited about your work?
Myself.
Good. So you need to sense that you are very excited with your finding, rather than look like something very usual, very routine.
Okay.
So the other thing is, when you pronounce, make sure you have the ending with the F, with the TH, and that will have never been done. All right. So even your heading, many of your heading, the English is wrong. All right. So S or without S. So this one you need to watch out with your heading.
Okay.
Okay. I want to ask a question. Let me ask Nan. Nan, can you turn on your audio?
Yes, Professor.
So you are not working on the Chinese medicines. I want to ask you, Kenneth mentioned about AR, ASR. What is this?
ASL.
AR, ASR. What is he referring to?
I don't know. Usually, I didn't catch up him properly.
Okay, so she doesn't understand Kenneth. All right. So, Alan, what is AR, ASR? Lim. Yes, you.
I think it's astragalus reticulum.
ASR.
Oh, AR is astragala radix. ASR is astragalus reticulum. I think I didn't catch the beginning of the
So
list.
Kenneth, you never give me a good nomenclature of AR, ASR.
Okay.
Is that important, Kenneth?
The language is very important.
This is important. Why?
Yes. Because AR or SL cannot.
Well,
some people
because these two half make up the Dongguai Bushu Tang, right?
Yes.
Okay. The other thing that I have some problem. In your presentation, you are talking about DBT and DBT lockout. So I suppose you are referring to the color coding lockout DBT, right?
Yes.
It's not DBT lockout. So you lock out DBT is 0. So this may be just a wording problem.
Okay.
But however, Kenneth, your study is more than that. Why? You have done a lot of work on the omics with the AR alone, ASR alone,
Okay.
AR plus ASR by simple mixing. So what this methodology implications? What are the implications? Are they important? First of all, Kenneth.
The borrowing effect is affect the.
Yes, it's a bonding together.
Bond together.
So you never talk about that.
Because,
am I right?
Okay. Yes,
so
yes.
You overlook that particular area. So if I were you, you should somehow also mention that that is one of your findings. Besides, lockout color coding.
Okay.
All right.
Okay.
So I think that your introduction of Dongguai Bushu Tang is too long.
Okay.
Your introduction.
Okay.
All right. And each particular slide, I'm not able to catch. For example, let's look at the some of those seahorse analysis. Just anyone. Okay, let me ask the questions. Xiao Xiao.
Yeah.
So what this slide is talking about, do you know? I know that you are late this morning.
Sorry. So about this slide, the metallic. Yeah, it is about the omics.
It is about omic, right? So what kind of omic it is? Proteomic? It's a transcriptomic.
Sorry, I'm not so familiar with this one.
I cannot hear you.
Yeah, sorry, I'm not so familiar with this one.
Okay, let me ask another one. Amy, so what kind of omic this is?
I can't catch up with Kenneth.
The what?
I'm not very sure.
You don't know, right?
Yeah.
So Kenneth,
I haven't done this before.
Kenneth, what kind of omic it is?
Plus omics.
This is not omic. This is a COS, right? Mitochondria bioenergetic analysis.
Yes.
So nobody understand.
Okay.
All right. So that particular, you give 2 or 3 slides for introduction, but none of that you can give me a clear answer. So look at the number 14 slide. Number 14, sub-say. Okay. So, and again, do you know what is the problem in here? Look, the one down here, 15. Okay. So I want to ask Gary, basal respiration. Do you see the basal respiration, the one in the middle?
Yes, he do not have the y-axis title.
The one on the top is what is this one?
OCR.
The one, the line, the curve with 2 stars.
You mean the middle?
You have the one in the center, basal respiration, right? You have a curve.
Yes.
So the upper one line. The one that you put open circle.
Yes.
What is this?
Wow. And the control, right?
So what about the flat circle?
Flat circle is 0.1 milligram per ml of
what?
I don't know.
So,
DBT.
Kenneth, do you know? Kenneth.
Yes.
So Gary do not know. I do not know neither. So what is this nomenclature?
The nomenclature should be for the right circle is the DBT with the TBHP treatment.
Okay, but where you have mentioned that one?
In the
it's on the bottom.
Bottom.
So again, you need to put opposite. You put the abbreviation first and make it bigger.
Okay. Okay.
Okay. So this is my comment, Kenneth. If you want to make the examination easy for you and get a pass, I will strongly recommend you go home and talk to the computer and record of your presentation. Did you do recording for this time, Kenneth? Did you record your presentation on the Zoom?
Let me check.
Yes or no?
Let me check. No.
No. Okay. So this is bad. All right. So I will recommend you to make more practice at home and present it to the computer and the iPhone so that you can hear what you have presented. All right. Watch out the English and to eliminate so.
Okay.
And make it more enthusiastic. So don't do it like and everybody will fall asleep. Okay.
Okay.
Look, slide number 1. Kenneth, can you speak the title in a very enthusiastic manner?
Mass spectrometry-based multi-omics technologies in resolving the action mechanism of the herbal medicine.
Okay, can you do it again?
Mass spectrometry-based multi-omics technologies in resolving the action mechanisms of the herbal medicine.
Okay, that sounds better. All right. So the other thing that I saw that for some reason you put many question mark on your slide here. Maybe can you show me some of the slide that you're asking a question?
Question mark. Okay, sorry.
Okay, so you don't need that many question mark. All right, Kenneth. So this is the
Okay.
way that I do it for you for a student. So when you do it for the examiner, one is enough.
Okay.
Okay. So the other thing that for number 4. Okay. So if I were you in this slide, I will put down the study that we have from Carl Jim's laboratory on the Dongguai Bushitong, the paper.
Okay.
All right, such as Guan et al. Blah, blah, blah. So you put it on the right hand side in this diagram. So and then on the left hand side, you don't have to put the name of the paper.
Okay.
And then I make it smaller. So and then you come out a conclusion. For all this year, your laboratory had done a lot of work on Dongguai Bushitong with the action mechanism to EPO, estrogenic antioxidations. However, the exact action mechanisms is still not known, particularly by individual chemicals.
Okay.
You understand what I'm saying? So therefore, make
Yes.
you that you are add value to what we have done before.
Okay.
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 All right. So maybe we can ask few questions. So let me ask Gao Song. You can ask Kenneth for any questions.
Okay, Professor. Kenneth, question 1. I find that you noticed you mentioned the qi research in the introduction part, but I cannot find this kind of research throughout your presentation about the qi research.
Okay.
Right.
Yes.
And I think the question number 2, you mentioned that the DBT is commonly used for menopause and other women diseases, but why you use female or use a male rat to do the animal study?
So what's the answer, Kenneth?
Okay. The what mentioned is cheese. We in the introduction we will do some definition. Is the cheese related to energy metabolisms? For the chapter 1 we will study the mitochondria bioenergenesis as well as the biogenesis. And the second question is because the sorry. For the why we use the bands is to provide the prevent the metabolic changes in the estrous cycle.
Kenneth, when
Yeah.
you speak English, do not look like speaking Chinese. So when somebody asks you a question, you think of the answer. So you make up the English sentence in your mind before you spit out. All right? You understand what I'm saying? So
Yes.
our response, I don't know what about you, normally that we respond into Chinese first and then translate it to English. So that may take time, maybe millisecond. So you transform that into English before you speak. Okay? All right, so answer the question, Kenneth.
And second one is to the second question is prevent the metabolic change in the estrous cycle.
The what? Metabolic change of what?
Estrous cycle.
Estrous cycle, right?
Estrous cycle.
Okay. Wow. So are you happy, Gao Zhong? Gao Zhong, are you happy?
Okay.
Okay.
Okay, I know that.
So, Xiao Sha, ask him a question.
Yes, I have two questions. The first one is about the yeast-induced inflammation in rats. Can you go to that slide? Yeast-induced inflammation. I just want to ask, the yeast-induced inflammation is a well-built animal model? I mean, why they choose yeast to induce the inflammation in the rat?
Yes. The first one will use the yeast induce the information is about related to the fever.
Kenneth,
Yes.
watch out your English.
Okay. The information is related to the fever. So, and this is a well-established models in, yes.
Okay, you mean the most inflammatory response is about the fever, the temperature of your animal, right?
Yes.
Oh, okay. So my second question is, I noticed your first part is about the maybe heart protective function of the DBT. Am I right?
Yes.
And the second part is about the bone proliferation and differentiation and something. So I just want to ask why from heart to bone, these two parts is any relationship between them?
Okay. The two part is the part one is to more to solve the mechanism about qi, which is related to energy metabolisms. And in the H-lyse to cell life, they have very high abundance of the mitochondria. And this is very good model to study the mitochondrial biogenesis as well as the bio and energetics. So this is to resolving the problem about the low qi. The second one is to solve the action mechanism about the roles of active compound in the glycosis. And we found that the butterfly effects in the osteogenesis model is much very significant after the glycosis knockout. So I'll use the bone model as our study model to be solving the roles of the action mechanisms of the glycosis.
Okay, thank you.
Okay, Tasha.
So I have about two problems. So first of all, in your introduction part, you mentioned the acetone wash of ASRs. So have you ever do any researches about the acetone wash effect on the DBT formulations?
You mean the ethanol wash, right?
Yeah, acetone wash.
In our lab, peers will do a lot of research about the wash with ethanols for the actual genetic synthesis radius, and they will remove the volatile oil in the ASR.
Oh, okay. And the second question is about the MMP assays. So what is the FCCP here?
Sorry, MMP. You mean the mitochondrial membrane potential?
Yeah, about 17, I think.
FCCP is negative control because the FCCP is the uncoupler to mess up the mitochondrial membranes. So this is a very well-established negative control for the mitochondrial membrane potential assay.
So Kenneth, this heading, DBT enhanced with a S. Okay, so mitochondrial bioenergetics related gene. So you have two adjectives. So that could be DBT enhanced bioenergetics related gene and mitochondrial membrane potential in cardiomyocytes.
Okay.
So this is the properly referring to what you are doing here.
Okay.
So yeah, Maggie.
Yes, I have a few questions. The first one is that why do you choose DBT formula to study the knockout, the chemical knockout study instead of other formula? And I also noticed that you picked the decoction described by Li Dongyuan. As far as I know, there are more than one decoction described for the DBT. So what's the reason you choose the one described by Li Dongyuan?
Okay. For the first one questions, because the DBT Dangbei is a very simple Chinese decoctions. It's just two herbs, the astragalus as well as astrogenesis radius. So, and they have a lot of
Kenneth.
Yes.
Dongguai is Angelica sinensis.
Angelica sinensis radius.
Radius. Okay, so Angelica sinensis radius.
Angelica sinensis radius.
Radius. Oh.
Radius.
Okay.
So, and in our laboratories, we have a lot of study about these formulations. So we'll choose this for our further study.
I strongly recommend you to cut the soap.
Okay.
So, okay.
Okay. And the second ones, DBT have a free formula utilized for the pharmaceutical research studies. So in our previous
so I think you're going to cut down the soap, Kenneth.
Okay. Therefore, we will choose this formula, the Dongyuan's formulas, because we would because this formulation is much better pharmaceutical effect compared to other two formulas.
So what is your question, Maggie?
My second question was, what's the reason he chose Li Dongyuan's prescription to study instead of other DBT prescription?
So Kenneth, I think number one, you already mentioned. So this formulation of Dongguai Bushitao has only two herbs, all right? So the other is more than two. Number two, this is most popular being used today. Do you get it?
Yes.
Bella.
Here. I have one question. Kenneth, from your data, I found the calicosine depleted DBT possessed much reduced activation in your study. So how could you make sure after the semi-preparative column there were no chemicals trapped within the column?
So Bella, this is a makeup question for Kenneth.
Yes.
No, I'm asking you.
Yes.
Okay, so Kenneth, during the examinations, the open sections, you need to ask maybe 4 or 5 your classmates to ask you some questions that you have backup slide.
Okay.
So Bella is one of them, right?
Yes.
Good.
Yes.
So what is the answer, Kenneth?
The answers previous studies. So we will do a lot of QC, qualitative control, and qualitative assurance of the chemical depleted methods. For examples, we will compare the isolated glycosin as well as the glycosin standard. We'll use the LC-MS to quantify and quantify the compounds, and the result of the molecular weight is similar. And the second one will use the MNR to study the substructure qualifications, and the result will show that the glycosin and the glycosin fraction is very similar to our results. And finally, we will the DBT was passed after passed through the HPLC column compared to the DBT and the DBT lockout glycosin. We found that the amount of the polysaccharide is very similar.
Got it. Thank you.
Kenneth, I think you are missing another key issue here.
Yes, I know the biological similarity. I have not this perhaps that I will.
So
yeah.
Amy Kong have done some of the work with HPLC DBT.
Yes.
So DBT go through HPLC column.
Yes.
So in this diagram, the substructural quantifications. So the upper one is color quotient, the lower is color quotient fraction.
Yes.
So I think Amy also do some of the work with the MMR between the Dongguai Bushitong and Dongguai Bushitong lockout color quotient.
Okay, I
So
check.
this is for small metabolite. So the polysaccharide do not change. The HPLC DBT do not change, particular from biological activity, like what I just mentioned to you this morning.
Okay.
So
I will add more detail at the belt slide in this area.
So you put everything in one slide instead of 2 slides. So those are diagrams. You can just get rid of them. Those are instruments.
Okay.
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 So what is the another questions? Maybe who else? Gaga.
Yes. Actually, I'm not familiar with this field, but I just want to ask a question because in the field that in the slide that DBT and the DPT with the Cargodius, right? I don't know. I read it right. If I read it right, maybe I make some slides, but why the DBT and DBT with the and the simple Cargodius have the different functions. The simple Cargodius was just have no function, and together with the DBT, the function will be more better. And why is it?
Well, Kenneth, I think Gaga do not catch what have you done, right? So she is completely misunderstanding your presentation.
Because I'm not familiar with
Gaga, I think if Kenneth can make you understand, Kenneth should get an A. All right. Unfortunately, Kenneth cannot either me or you to understand what he's talking about. So Kenneth, so this is important, particularly in the beginning of the study. So you emphasize that you like to compare Dongguai Bushitong, compare Dongguai Bushitong without color quotient, compare Dongguai Bushitong with single herb, compare Dongguai Bushitong with AR but ASR by mixing together without boiling. So this is your key objective in the omic analysis.
Okay.
Am I right, Kenneth?
Yes.
Okay, good. So Kelvin.
I have a question. Kenneth, can you show me the result of the macro mass? No.
Oh, okay.
So Kenneth, slide show.
Yeah, this one. So this one, you say that the DBT color-coding knockout DBT has much more effect than the DBT only, right? On the differentiation of this micromass model.
Yes.
Does that mean DBT knockout has a better effect than the DBT?
DBT is sorry.
The knockout color coding. Do they look like more than that than DBT?
The knockout, the color-coding knockout DBT has a better effect than the DBT in the induce the differentiation of the micromass model from this result.
Okay. Okay.
Because is that
because,
right? But
yeah,
because I saw the results from the part 3 of the presentation. In the omics result, you say that the knockout DBT has a weaker effect in inducing the differentiation of the bone, right?
Yes.
Are they controversial?
You mean the osteogenesis? So
yeah.
Okay.
Kenneth, the slide in here, I think you need to make it better. Number one, this slide, which part is the most important part?
DBT and DBT log.
No. The most important part is the distinguishing of the micromass, right?
Yes.
So, however, so on the left, this is occupy 1/3. On the right, only the naming of those Asian blue and the Alcindrine red, you occupy another 1/3 of the space. So the space wise, I will think that the middle one to make it bigger.
Okay.
You understand?
Yes.
All right. So those things you can put on the bottom. So what is the concern is you make the circle much bigger.
Okay.
So because Kelvin is thinking the DBT as you shown it in here, they are more dense with those red spot or less dense as compared to DBT delta color coding.
Okay.
However, Kelvin's impression may not be correct because the DBT case, the red spot, they are all centralized in the middle. Okay. So when you make it bigger, so you can you 你可唔可以放大俾我睇下呢度？ So in principle, yes, go. Okay, look at the okay. So in that case, that may be better because this diagram, Kelvin, the delta color quotient should be less red color than the DBT. Am I right, Kenneth?
Yes.
So this is a kind of a misrepresentation of what you are presenting.
Okay.
So like Maggie always say that when I look at Michelle, she always say that this is a statistically n equal to 5, all right? But unfortunately, she will show you that the one is opposite. You understand? The green one is okay. The blue one is okay. It's very significant that the DBT is better than the bottom one. So, but on the DBT one, so the red dot, they are all localized in the middle. That makes us an impression that they are better than the delta color quotient is better than the Dongguan Bouchard.
Okay.
So I don't know whether you have a better figure or not. If you have a better figure, well, you always say that n equal to 5, right? So there should be another representing diagram.
Okay.
Is that right, Kevin?
Yeah.
Is that what you are thinking?
Yeah.
Any other questions, Kevin?
No. Thanks.
Nan, do you have questions?
Yes. I heard a lot you talk about PCA model, right? So I see the almost the last slide, and you show about the PLSDA model. What is difference between these two models?
Okay.
Because I have
what are
no
the differences
idea.
between the two models, Kenneth? PCA and PLD, PLSDA.
PCA is full of principal component analysis. This is unsupervised model to unsupervised to use the just pure machine learning approach to separate classification the two control or experimental group. And for the PLSDA or OPLSDA is supervised model to assign control group and the experimental group manually. But in the metabolomic studies, this is a very important tool to find more the features or find more metabolites between the control or experimental groups. So we found the biomarker, we will use the OPLSD or PLSD model.
So explain them in a more layman term. So the PCA is a random process. You input every data that you have, right?
Yes.
So the PLSDA is
assigned by human.
So that means that you only pick up those one that you think of interest.
Yes.
So the OPLSDA is
this same gives same functions, but the OPLSD if compared with just two group, for example, just control or experimental group, is much better.
Okay. Are you happy, Nan?
Yes.
Odin.
Kenneth, I have a question. Do you know the role of calicosine in 当归补血汤 in terms of molecular target?
So this is you know that the glycosin in the glycosin for the osteoblast is less or not any effect, but based on our omics result, we would like to predict the molecular targets. We have two reasonable results. The first one, the glycosin may change the membrane structure and facilitate the action of active compound of DBT. The rotation freedom molecules is increased in the large ordered membrane. Suppose note because the sphingomyocyte or sphingolipid was changed in lipidomic results. So this lipids was shown. Yes.
This is may not be right because this is what you proposed by your own analysis.
Yes.
But I think Bodhi is asking, what are the targets have been known today for calicosine to add on?
Okay. This is the literature is solid result for the to answer these questions. Yes, stimulate the IGF.
No. So if you say this is they add on the IGF-1R, have you done experiment that they activate on the IGF-R?
Omics result is measured.
Not omic. Omic is a kind of a prediction.
Okay.
So did you look at Carl Jim's paper in 当归不是汤?
Yes.
So we have 1 or 2 students have been working on the calicosine, including you.
Yes.
So what is the target that we know that calicosine have been working on?
The estrogen receptor or
yes, this is estrogen receptors, right?
Yes,
Have
when
we
pathway.
demonstrate that they act on the estrogen receptors.
Okay.
This
And,
is well known.
Yes.
But on top of that, what you predict?
It's by previous
You are
lab.
predicting 1 and 2 here, right?
Yeah. And the second one is
Is that what you want to ask about it?
Pardon?
Yeah, is that what you want to ask? What the color quotient target is?
Yes.
So there are two kind of a statement, Kenneth. Number one, have been known. All right, this will be the estrogen receptors.
Yes.
Who did that?
Right.
Huh? Who did
Estrogen
that?
receptor.
Well, some of the people have done that long time ago. All right.
Right.
Yeah. So what about the predicted function?
The
predicted. So by omic analysis. By who?
Me.
Good. By Kenneth Kwan, right?
Yes.
So you got to make sure that which one is have been known, which one is a predictor analysis.
Okay.
So any question about it?
No.
Okay. Maybe we ask Wai Yao. Wai Yao, do you have questions?
Yes. I have two questions. Question one, for the energy metabolism, I want to know the introduction for the equipment. And question two, for the activity of AR, ASR, DBT, and AR ASR, I found that the AR ASR, the activity is lower than the single herbs. Can you explain the reason or discussion the questions? Thank you. Kenneth.
Yes. You mean the introduction part or?
For the energy metabolism. Page 12. Yes.
Yes. Okay.
Perhaps I don't know the how did it works?
You mean the seahorse or?
Wow, it's a seahorse, right, Kenneth? Nobody know what is the seahorse talking about. So including Wai Yao, I think including others.
Okay.
So you better make well, you have 3 slides talking about the seahorse, but nobody understand what you're talking about.
Okay.
So how to make that clear?
Back the face to more explanation about the seahorse principle.
Wow, you already have 2 slides or 3 slides talking about explanation. It's the one who explained the problem is not clear.
Me.
Yes, it's you. All right, make it simple.
Okay.
So what is the second question's answer?
The activity of AR, ASR, DBT, and AR ASR.
Well, look at the mitochondrial morphology. So I think this is why I was talking about those lines.
Mitochondrial morphology.
So okay, so what is this?
16, page 16.
Okay.
So he is referring to AR, but ASR, why that is different to AR or different to ASR?
You mean the gene analysis, right? Or mitochondrial potential?
For the healthy cell model, AR加ASR is lower than AR and single ASR. You know.
Oh, I got your meaning. In the two models, the health cell model or weak respiration cell model, the AR and the ASR, the response is similar. However, we find that a uses things why the ASR, the AR plus ASR, the response is two different, right? Is that you ask these questions, right?
Okay. Yes, what's the answer?
The answer is I don't this model will need to more clarify or more study.
That means you have no answer, right?
Yes, no answer.
But I think that you did some of the work, if I recall it correctly, in your omic analysis. I don't remember which omics that you mentioned that the AR but ASR, they are disturbing the whole system at all.
Yes.
As compared even to the single hub.
Yes.
Could that be an explanation? Well, first of all, the AR but ASR, you separate the two out without binding together. So which pharmacologically is not as good as 當歸不是湯.
Yes.
Right? So on the same time with the lab pharmacology, I remember you showed me that they are more disturbing as compared to a single hub.
The
That slide?
Yes.
Show us.
I've loaded this slide here. I will add this slide. Yeah, soon. Yeah.
Speak louder, Kenneth.
At this moment, I have not these figures, but okay. I will prepare soon.
Okay. So any other questions?
Yes, I have a question.
Yes, Evan.
Kenneth, you do a lot of mass spec for your metabolomic study. So can you explain the use of different mass spec techniques like orbit trap and other?
So what is the MS? So can you tell us?
Okay, we will use the orbit trap and time of flight mass spectrometries. Then I have a backup schematic diagrams. And for the orbit traps, after the ionization of the chemicals, the chemical will go to the C trap for the ion concentrations, and they have a lens to guide the ion to the orbital traps. This is the ionization, and this is this is the mass separators, and they will provide different helps and oscillate if this mass to charge ratio is fulfill the oscillations, the molecule will stable and go to a detector. This is the working principle of the orbital stress. For the second one, we'll use the time of flight mass spectrometries, and the molecules will, after ionization, will go to the time of flight tube. After in the extraction after in the extraction electrolytes, the kinetic energy will become the equal or become same kinetic energies, and after this time of flight tip, they will depends on the mass. If the mass is very mass is very light, they will go to the detector faster. So this is the separation mass mechanism of the time of flight mass spectrometry.
So are you happy, Evan?
Yes, thank you.
So Kenneth, I think to summarize the today meeting, I think number one, you need to practice more at home. Kenneth.
Okay, yes.
So sounds like that you never did practicing at home regarding your presentation.
Okay.
So if I were you, I will record it on the phone, and you can hear what you just say.
Okay.
So revise the PPT, make it more indicative. So put down the nomenclature, put down the abbreviation. So take out the ABC, all right? Put the heading.
Okay.
So watch out your English.
Okay.
Okay. Be speak slowly and pronounce the 美音 as good as you can.
Okay.
Right? With ED or with S, that kind of thing. So that will slow down your English and so that people can understand better.
Okay.
So the other thing is, if I were you, because we'll be on the Zoom, so you can have print out those backup slides on a table, print it out right in front of you, right?
Okay,
Possible,
like this,
right?
Yes, like this. Okay.
So and also you need to find the backup slide as soon as you can, rather than we spend a few minutes to find the backup slide.
Okay.
So who is going to help you to ask few questions for you?
Kelvin.
Okay, Kelvin, are you here?
Okay.
So
okay, no problem.
Who else?
Brody.
Brody, it's okay?
Okay.
So you already have Bella, right?
Yes.
Well, who else?
Gao Song.
Gao Song, okay. So make sure that they ask good questions. And normally, we will ask a question that you have not presented here, so that you have backup slide.
Okay.
Okay. So, well, one more reminder. I think your presentation is not very focused. It's not emphasizing particular with the slide that you have and particular with the experiment that you have. All right. So that's all I have to say. By the way, Kenneth, the acknowledgement. Can you go that slide? So I will recommend you acknowledgement. So beside those names, you should put down on the bottom, well, some of the paper that you publish.
Okay.
All right. So because this is a requirement that you need to publish papers.
Okay.
So put down that at least those are paper with your first offer.
Okay.
And with those are being submitted as well.
Okay.
Okay. So that's all I have. Any other questions? Okay. If not, bye-bye, Kenneth, and good luck.
Thank you, Professor.
So by the way, make sure all those students, they come in to your seminar.
Okay.
All right. Okay, bye-bye.
Okay. Thank you, Professor.
Bye-bye, Wai Yao.
Thank you.
Bye-bye. Chat.